One of the more prominent areas of research is to understand how our gut can impact our brain health. And in particular, changes in the composition of our microbiome have been associated with mental illnesses, in particular depression and schizophrenia. And the thought is that by regulating our gut health through the regulation of these bacteria that reside in our gut, that we can treat mental illness. And in fact, a recent study that was published in Nature Communications from Erasmus Medical Center in Rotterdam suggests a possible mechanism by which that might occur. What this group found was that the bacterial microbes in our gut could actually impact behavior potentially by synthesizing a bunch of molecules known as neurotransmitters and other metabolites. These include things like glutamate, butyrate, serotonin, and GABA. What's interesting about this study is that those neurotransmitters and metabolites have already been directly implicated in things like depression. So the thought is now that one possible way in which our gut microbiome can impact our behavior is by synthesizing these critical molecules. And this study really does, I think, add to this growing body of work linking our gut health to our brain health in meaningful ways. A complementary study out from Amsterdam University Medical Center, also published in Nature Communications, explored these mechanisms further. The gut microbiome is really a collection of many different bacterial microbes. It's thought actually that it's estimated that nearly 1 trillion bacteria reside within our gut at any given time. And it differs in terms of its composition across individuals quite dramatically. What this group found was that not unexpectedly, gut microbiome differs or varies across ethnic groups. Given the fact that your gut microbe composition is greatly impacted by the types of foods you eat, this finding was not necessarily all that surprising. But what was important about this study was that it didn't matter in terms of its association with depressive symptoms, meaning that disturbances in the gut microbiome, independent of ethnicity or differences in composition, still seem to be strongly associated with depressive symptoms in these patients. And this is an important finding because there's been a huge push to develop therapeutics that target the gut microbiome. For example, administering or treating an individual by treating their gut microbe composition. And this study, I think, importantly shows that strategies to regulate the composition of our gut microbe may be effective across ethnic group, irrespective of slight or subtle differences based upon either their environmental, i.e. their diet-related differences, or genetic composition. Both of these studies are directly related to the work that we're doing at the Brain Body Research Center at Mount Sinai. For example, we're looking at these interactions between brain stress and anxiety centers and gut health. The gut is an important site. It's an important immune organ, in fact. It has one of the largest populations of immune cells, and it's really thought that those gut microbes are interacting directly with the immune cells there. And depending upon the composition of your gut microbiome, it can have either positive or negative effects on your own immune health. One of our recent studies has been examining connections between stress centers in the brain and the gut microbe environment, including how the perception of stress in our brain impacts gut health through a microbiome direct effect on the immune system. And we think that ultimately, by understanding these mechanisms, we may be able to develop better antidepressant therapeutics and treat depression in more meaningful ways. I'd like to thank all of you for joining me today. It's been really fun talking about our research and the really exciting research that's going on in the field that's identifying novel ways in which our body is coping with stress, and hopefully will lead to a new class of therapeutics that target our brain and body interactions.